Uh, what are proteins? I think this is quite simple as a question, but good way to introduce the topic. So they are large complex molecules that are found in cells and organisms. Their structure is dictated by DNA, right? So transcription, translation into a polypeptide chain. Their structure contains carbon, hydrogen, oxygen. So this is like lipids and carbohydrates. The only different thing is that they also have nitrogen. And so the major source of nitrogen comes from is our proteins basically. So proteins are made from 20 different amino acids. These amino acids are dictated by the DNA, the codons that lead to the amino acids. And so there's 20 of them. In nature, there's more than 20 amino acids, but only 20 of them are uncoded by DNA. And so when we're talking about proteins in terms of food content and in terms of energy, each gram of protein provides 4 calories with a big C of energy. Okay. The amino acids are the building blocks of proteins. There's 20 different amino acids that are found in proteins. These amino acids are going to be either hydrophobic, hydrophilic, charged, not charged, and whatnot. So this you've already seen in biochemistry. Their composition, usually you have a central carbon atom with an amine group, so an NH3, NH2, an acid group, so your carboxyl group on the other side, and a side chain. And so in the diagram, this looks like this. So you have your amine group, your Na2 that I just told you about, bound to a central carbon, which has a carboxyl group on the other side, COOH. You have a hydrogen atom bound to the carbon as well, and a side chain. Now this side chain varies for each of the amino acids, and so you'll have different properties based on this side chain. If it's charged, then the whole amino acid will be considered charged. If it's hydrophobic, rings, whatnot, rings like tryptophan, for example. So this side chain is what gives the property to the amino acid. And depending on the property that the amino acid has, it can help in the structure of the protein, right? So when we talk about amino acids, we can talk about essential amino acids just like essential lipids. So this means that you need them in your diet, and so you must obtain them from food, and they cannot be produced by our body. If we're talking about non-essential amino acids, these can be made in our body, and they're usually made from essential amino acids through a process called transamination. So in your body you have these keto acids that can be turned into amino acids by the transfer of the amine group. And so if you have the essential amino acid, too much of an essential amino acid, and you have the keto acid that has a similar shape and form as the non-essential amino acid you're trying to make, well, you can transfer an amine group from here to the keto acid and produce your non-essential amino acid. Here's a list of the essential versus non-essential amino acids. The essential ones, you have 9 of them, and the non-essential, you have 20. This means that you can make all these from these, right? Because there's a transfer of the amino group to whatever keto acid that resembles these, okay? As in diagram, if you have a keto acid that looks like this, then you can take a amine group from valine. If you have too much valine, transfer it to this keto acid, and suddenly you're producing glycine. Valine is essential, glycine is non-essential. Okay? Very simple, and you can imagine different structures. For example, you have valine here, and you have a different side group here. Let's say, I don't know, this one, then you'll be producing leucine here. Okay? Additionally to the essential and non-essential amino acids, there are also those known as conditionally essential. This means that they are non-essential in reality, but because your body cannot produce them, then they become essential for you. Okay? For example, in a disease called phenylketonuria, you have phenylalanine that gets accumulated in the body because it cannot be converted into tyrosine. Now, tyrosine is a non-essential amino acid, but because we cannot make it because of this mutation that you have in an enzyme that produces tyrosine in your body, then tyrosine becomes essential for you. Additionally, your diet also affects what's conditionally essential or not. Vegetarian diets, for example, are not rich in methionine. And because they are not rich in methionine, then you'll have an unbalanced level of cysteine. And so cysteine becomes conditionally essential for you. Okay? So this is what it looks like. You have phenylalanine that comes from dietary food. It gets converted into tyrosine. So even if your food doesn't have tyrosine, you can make it in your body through this reaction. And so tyrosine then gets converted into melanin. Melanin gives you your pigmentation in the skin. If you don't have this, then you're not going to produce melanin and you'll have albinism. Tyrosine is important for many proteins, including many proteins, and then tyrosine also is important for the production of dopamine. And so you can imagine that phenylketonuria might lead potentially to delayed cognitive development, because dopamine is important for learning, for memory, for reward system, and whatnot. Okay. This is just a more complex diagram of what I showed you right now to show you where mutations can happen. So obviously, if you have this mutation in the enzyme that converts phenylalanine to tyrosine, then you'll have an accumulation of phenylalanine, and phenylalanine is toxic in high levels. It can cause brain damage and whatnot. Tyrosine, if you make it, then you need to also convert it into dopa to make melanin. If you don't convert it into dopa, then you end up with albinism. And here in this case, if you do have this enzyme and you make tyrosine, right, so you'll make many of the intermediates, right, required for development. But if you end up having a mutation in this enzyme, for example, which converts this HA acid, then you'll end up with a variation of phenylketonuria called alkaptonuria, and you're basically accumulating this acid here. Okay, so this is why it becomes kind of essential. You don't need it. Your body can make all this just from this, but because now you have a mutation here, suddenly you need supplementation in some amino acid that is non-essential to have all these reactions happen. Okay, and if you don't, health problems. Same thing with the cysteine that I told you about right now. Cysteine is made up from methionine, and there are many other amino acids that are produced from methionine. So if you don't have it, you end up not producing homocysteine, cystathionine, cysteine, taurine. Taurine is an amino acid that you'll probably find in high levels in energy drinks, for example. It has some benefits for energy and whatnot, but it's also very important to produce bile salts. Okay, and so if you don't have methionine, then you end up in a deficiency, and so that makes everything else essential right after it. Okay, this is just a list of amino acids and their roles, just for your curiosity. I'm not expecting you to learn all this by heart. There's Google for that. It's just to give you an idea of what they do in the body. Okay, how are proteins made? As seen in biochemistry 1, there's amino acids that are just linked together through a peptide bond, and the peptide bond happens between the carboxyl group of one amino acid and the amino group of the other, as you can see. So you have your amino acid number 1 right here with the amino group, the side chain, the hydrogen atom, and then here you have that carboxyl group which interacts with the amino group of the other amino acid, forming a dipeptide bond, so a peptide between two amino acids. Now, these proteins are made from your DNA. So your DNA is what will provide the template for the proteins and tell your body which proteins to make. You don't make proteins like you make lipids, right? So lipids, it's the same lipids everywhere, okay? And so when you eat it, the same components will be used to make those lipids, okay? So if you eat triacylglycerides, well, the triacylglycerides will be broken down, and then the components will be used to make triacylglycerides in your body, okay? Proteins, they have a template, and the template comes from your body, because it's not the same proteins between, if you eat, for example, a protein that comes from cow, for example, they don't have the same proteins as us, while lipids, it's pretty much the same thing. And so those proteins will be broken down into their amino acids and then used to rebuild proteins that are specific to humans, okay? And so this comes from our DNA. DNA has the code to produce the proteins that are specific for us, and this DNA will be transcribed into a messenger RNA, which will go to the ribosome, leave the nucleus, go to the ribosome, and produce a protein through translation. If you need a refresher on this, there's the diagram right here. So you have DNA producing mRNA, messenger RNA, which will go outside of the nucleus, be bound by a ribosome. The ribosome reads this code and produces a polypeptide chain from it. And the way this works is that each codon that you have on your RNA will be specific for a tRNA, and the tRNA is just a molecule of RNA that carries the amino acid. And so tRNAs will come, will drop the amino acid, and will leave, and then another tRNA comes and brings another amino acid, and then just follows the code, right? When the polypeptide chain reaches, when the ribosome reaches the stop codon, there's the reaction where you lose the polypeptide and it's released, okay? Now when it's released like this in the cell, it needs to take its shape, right? It cannot just stay as a long strand of protein. And this happens through a secondary, tertiary, and quaternary structure that we'll talk about in a second. But before that, I want to mention an important concept. Proteins get made and degraded in a dynamic process. Some proteins get degraded more often than others. Some proteins that are very important and cannot really be recycled will stay in the cell for a longer period of time. So their lives are not all the same. Ribosomal proteins, for example, will not get degraded as often as a heme protein, for example. And the reason for that is some of them, they need to be always there, and so they will always stay there, okay? This concept is called protein turnover. So proteins that are degraded, proteins in the cell will be degraded, and they will provide the building blocks for new proteins. So this is the protein turnover. This creates an amino acid pool that contains both the amino acids from food and from cellular breakdown. So the endogenous and exogenous amino acids, right? Those that come from food and those that come from breaking proteins that are already found in the cell. This will produce your amino acid pool. Then new proteins are made from this pool here. And so the amino acids that you eat and the amino acids that get broken down from proteins already found in your cells are reused, are first pooled together, and then they're reused to make other proteins, other proteins like enzymes, antibodies, and whatnot. They produce also non-protein components that contain nitrogen, for example, serotonin, which is made from tryptophan in your gut. And so serotonin is a nitrogen compound, but it's not a protein, it's a small molecule. The remainder of certain amino acids that are in excess will be used to make fat. They'll be used to make energy, right, from gluconeogenesis, etc. Okay? The excess nitrogen that happens in these reactions, because nitrogen will be released as the reactions happen, will form ammonia, okay? And this ammonia is converted into urea in the liver, and urea is excreted in the urine via your kidneys. Okay, so urea is what eliminates the excess nitrogen in your body. And this comes from the reactions that happen in your body that use the amino acid pool to produce X, Y, and Z, and whatever is left, because the ammonia group here is released from these reactions, it will be used to form urea, and urea gets excreted. Okay. Question 1. If a vegetarian does not eat complementary proteins in a meal during the day, where does the body get the needed amino acids to synthesize proteins? Well, if you are lacking some amino acids in your diet, your body will basically get those amino acids from the amino acid pool. Because remember, the amino acid pool contains both the amino acids you get from your food and from the breakdown of other proteins in your body. So you were not born vegetarian, but probably you become vegetarian at some point in your life. So your body already has certain proteins, has all the amino acids in it, right? So if you don't, if your diet is good in terms of complementing whatever you're missing in amino acids from the vegetarian diet, then you'll be okay. But if you don't complement, your body will start breaking down its own proteins to complement your diet, and so to get the amino acids that are missing. So you can see how this can be quite problematic if you do it chronically, because your body is breaking up your own proteins to replace others. Okay. So as I said a few slides ago, proteins are organized into 3D structures. The primary structure is just a sequence of amino acids. It's the peptide that comes out from the ribosome. It gets converted into a secondary structure, usually by itself, by forming hydrogen bonds between the amino acids and sulfur bonds to create the disulfide bridges that you probably learned about. And this will make what you know as alpha helices and beta sheets. Now, these secondary structures, you won't only have one of them, you'll have multiple of them. So the combination of secondary structures in a polypeptide will form the tertiary structure. And this is where you have function that starts to happen. For example, if you're dealing with a channel that is transmembrane, usually you'll have a polypeptide in the primary form. Then that polypeptide will form kind of a alpha helices, and you'll have two alpha helices along, like separated by some sort of strand, and so that's your that's a kind of a tertiary structure, right? Because you have two alpha helices together bound by something, and so this will give the shape of the channel, which will have the function of carrying things through. So the function of proteins is carried by tertiary structures, right? You can think of active sites, phosphorylation sites, and whatnot in the protein. So these require a tertiary structure, which is a mixture of many secondary structures. Hopefully that's clear. Quaternary structure is when you have multiple polypeptides bound together. So multiple tertiary structures bound together will form the quaternary structure. What I mean by that is, if you take a look here at actin, these actin filaments, in this case here, this is a dimer. So it's two actin proteins of a tertiary structure that then interact together and form this dimer here. Okay, this is a quaternary structure. Good. So proteins have two shapes. They have long shapes and globular shapes. The proteins that are in shape of a long strand are usually found in muscle fibers, and they're important for contraction and relaxation of the muscle. Globular proteins are more flattened, like red blood cells, for example. They give this shape to the red blood cells, actually, and they provide flexibility so that these proteins can travel through the capillaries, right? And this is the globular shape of a protein. Microfilament is not globular, it's a long protein strand. This form also gives the ability of the heme to locate itself within the protein and carry the iron that binds to the oxygen and carries the oxygen. One characteristic of proteins is that they get denatured, and so they do lose their shape, which makes them lose their function. This is why when you put eggs in your pan, it starts changing form and shape. This is because of the proteins being denatured in it. But the primary structure is almost never lost. Okay. You can technically lose the primary structure if you're using enzymes or whatnot. You can break it. Okay. But the primary structure does not get lost in denaturation. It gets lost in digestion. Okay. And so things that can damage the secondary and tertiary and quaternary structure of proteins is heat, for example the egg, acid, base, so acid like HCl in your stomach. Heavy metals can interact with the protein and break its secondary and tertiary structure. Alcohol can also affect the shape of these proteins. And so for example, in your stomach, when you're eating food, the first part of digestion of proteins, well, not the first part of digestion of proteins is the pH, the low pH, the high levels of HCl denaturing the protein to make it more accessible to the enzymes that will break it down. Right? So this is it. Additionally, protein denaturation can be quite dangerous, especially when you're sick and you have fever. This is why it's important to control body temperature, because high temperatures can lead to denatured enzymes, and this will affect your body's functions. Okay, above 40 you start having issues. 41, 42 can be fatal for you, and it's just because the proteins cannot work anymore. They are getting denatured, or because of your pH. Drinking too much alcohol can decrease the blood pH, and by decreasing the blood pH, you end up with a lot of problems. This is why drinking too much alcohol is not good for you. The sequential order of amino acids in protein is called the primary structure. This is just basics. Now, when you're making a protein, you need the protein to be completed. You cannot have like a half protein and still have function, because proteins are associated with function most of the time. And so if you have an incomplete protein, because you don't eat all amino acids, or you don't have all the available amino acids, you'll make a low quality protein, an incomplete protein. And this is useless most of the time and will get digested and broken down as a repair mechanism, kind of a recycling mechanism. So this is not good. So you need a diet that is rich in amino acids so that it has the essential amino acids to produce a complete protein. Okay? Imagine you have a heme that you're trying to make, but you're missing one of the essential amino acids. Well, maybe it's the amino acids that's going to bind to the iron, right? And the iron element. And because you don't have it, then your hemoglobin is kind of incomplete and useless. And your body will just break it down. It's useless. There's a concept called complementary proteins. So again, when you're eating, and this is usually when you're following some sort of diet, sometimes you'll need to eat different foods to supply for all the essential amino acids to avoid building incomplete proteins, right? Because if you're going through the ribosome is going through the mRNA, and then suddenly it needs a methionine or whatever essential amino acid, okay, valine, and your body does not have valine, then you're not building that protein. You cannot add to it, and so the tRNA will come without a valine and leave, move next, right? This is going to break the whole translation process. And so a good way to have all the amino acids that are essential for you is to eat different foods that combine that supply the essential amino acids. Okay, this is just an example. If you eat, for example, legumes like lentils, chickpeas, and whatnot, are not rich in methionine and cysteine, and so the complementary food to that would be grains, nuts, and seeds. This could cover this limitation here. Grains are limited in lysine, so legumes are rich in lysine, and so you get the idea, right? So just this is an example here for you guys. Okay, that's it for structure and function. So now let's talk about digestion. So when you eat proteins, they'll come to your body and begin the digestion in the stomach. Hydrochloric acid will denature the proteins, as I told you, and activate your first enzyme that breaks down proteins, which is called pepsin. Pepsin will break down the proteins into shorter polypeptides, and these polypeptides will then be broken down further when they reach the intestine, right? The hormone that controls the level of hydrochloric acid production and pepsin release is called gastrin, right? Remember this from lecture 2. Now when these polypeptides and proteins that are still undigested reach the intestine, the small intestine, there's part of the pancreatic juices are proteases that will digest whatever is left. So you have trypsin and chymotrypsin, which will break down the tertiary, quaternary structure of proteins and can even break down primary structures. You have the intestinal proteases, which will break smaller peptides. So these are, for example, aminopeptidase and carboxypeptidase. And as you can see in the name, peptidase means peptide, not protein. So they will break down short polypeptides into a single amino acid unit. Okay, aminopeptidase probably breaks the peptide at the amino group. Carboxypeptidase will probably break the peptide at the carboxy group, right? Simple, makes sense? Probably going to have to listen to this recording at 0.75x. Going a little bit fast because too many things, but anyways. Now, when these proteins are broken down into single amino acids, dipeptides, and tripeptides, they will be transported into the enterocyte. Okay, and these enterocytes, they will be transported through shuttlers and transport proteins, which I'll show you in a second. But it's important to know that high doses of individual amino acids can lead to toxicity, and I'll explain that in a second. But there is a limit to how much protein, individual amino acids you can take. Okay. This is why BCAA, for people that work out, for example, branched chain amino acids, it's important to not take too much of those because they can cause problems for your kidneys and other stuff. Okay, so one thing that I want to talk about is chymotrypsin and trypsin. So in your gut, you have these two enzymes, and they're in the form of the zymogen, and that means they are inactive enzymes. The first enzyme to be activated is trypsinogen. It gets activated by a kinase on the surface of the villus of the small intestine, enterocytes, which will activate trypsinogen and transform it into the active enzyme trypsin. Now trypsin, as its levels increase in the intestine, they will activate chymotrypsinogen by breaking down part of it, which makes it inactive, to transform it into chymotrypsin. Okay, and this is important because you want to control, these are proteins. Okay, that's the first thing. So you want to control their digestion, because trypsin itself can break down these, right? And so you want to make sure that you are activating these enzymes at a specific time to avoid over digesting the enzyme and breaking them down and then not having any digestion. Hopefully that's kind of clear. So this is meant to regulate the breakdown of these proteins by themselves. I think this I talked about. Whatever is dipeptide, tripeptide, and single amino acids will be carried to the liver, which then will do a bunch of things to them and transport them. Okay. Now, Transport of amino acids across the enterocyte membrane happens in a similar fashion to carbohydrates. If you remember when I was talking about carbohydrates, I was saying that glucose requires a sodium transporter. Well, certain amino acids also require a sodium transporter. Others require facilitated diffusion. So this is analogous to fructose, for example. And so in the liver, they're modified and they will be distributed. Okay, and then they'll be sent to the muscles, for example, to build muscle. So the three modes of transport of amino acids: if it is a dio or tripeptide, usually it goes through endocytosis, okay, where the cell just engulfs whatever it's trying to take, and then carry it inside. Amino acids can get transported with a sodium transporter. Sorry, not dipeptide and tripeptide, bigger peptides. And then single amino acids get transported by a sodium transporter, which carries one sodium atom inside, ion inside the cell, and one amino acid with it. And then di and tripeptides are co-transported with a hydrogen ion. Okay. If we zoom a little bit here, you can see that you have the hydrogen ion being carried inside the cell, and the peptide will follow with it. And this is just to provide kind of the energy required to transport this. Okay. Obviously, when you have the ions going inside the cells, they need to be recycled, and that means they need to leave. So you have your sodium, your sodium hydrogen transporter. So one hydrogen atom will be kicked out, one sodium ion will go in, and then that sodium ion has to leave, right? If it accumulates too much, so then you have your sodium potassium pumps, and it's just a dynamic cycle. Okay. The function of proteins, I talked about it in the lecture. Just, you don't need to learn them by heart, but at least understand that there are many functions, and be aware of them. So you can read this on your own time. This is just a few examples talking about the same stuff. There are many types of proteins, and these proteins have different functions. So you need a highly diverse group of proteins. This is why the genetic code will code for these. So it's not the same proteins that you necessarily, let's say for example you eat a plant, whatever plant, well, the proteins in it are not necessarily interesting for the human body, right? So they will be broken down and rebuilt based on what's needed for the human body, and this comes from the DNA, right? And so these are the different types of proteins, and then their roles, and a few examples. One of the major reasons proteins exist is that they serve as enzymes to carry reactions, transform one precursor into a product or whatnot. So this is just a simple diagram that shows this. We'll be delving deeper into this during our metabolism class. They can serve as membrane proteins. Sodium potassium pump looks like this. Carries 3 sodium inside for 2 potassium outside, and just one movement provides the energy for the movement of the other in the other direction, right? Let's see. A patient with stomach cancer needs to receive her nutrition via a tube that bypasses the stomach and goes directly to the jejunum. How will the protein in her diet need to be changed? Well, in the stomach, you break down the bigger protein structures into smaller protein structures, and you denature the proteins also. So if you cannot do that, that means the proteins will reach your intestine as quaternary or tertiary structures. And so for that, it would be better if you can give, if the diet is changed into dipeptides and tripeptides than whole proteins, because as you reach the intestine, usually you have more of the carboxypeptidase and aminopeptidase to break down short amino acids and then transport them. You don't have time to denature and to break complex proteins into smaller proteins, because that happens in the stomach. In the intestine, it's the last small little peptides being broken down into smaller units and absorbed. You're absorbing in the intestine, so you want to have di and tripeptides as a replacement for her diet. If you keep whole proteins, they don't have enough time to be broken down by HCl, pepsin, chymotrypsin, and then trypsin, which are found right in the duodenum of the intestine, and so they will not get absorbed properly and it will get excreted, right? So this is important to take a note of this. Protein quality, so obviously certain proteins have a better quality than other because they contain the amino acids that you are interested in. There are 3 ways to determine protein quality. The biological value is basically the proportion of the absorbed protein from your food that is incorporated in body proteins. So this tells you how valuable a protein is, right? Because not every protein produces amino acids that your body will use and incorporate in other proteins. So this is the biological value. You can also take a look at the in terms of weight. So you can look at the gain of the weight gain of a growing animal compared to the protein intake, right? And that tells you the protein efficiency ratio. In other words, it's another way of saying how much of those proteins that you are taking are actually adding to your weight. Okay? And the last component, the last value is called the protein digestibility corrected amino acid score, and this refers more to the amino acids. Okay? And it is basically the amino acid requirement of a 2 to 5 year old child. This group is the most nutritionally demanding age group, so it's used as a reference group. Okay? So yeah, so that's pretty much it. In the exam, I'm not asking you to calculate any of these, so this is just some values that are used in nutrition to assess the quality of a protein or of certain amino acids. Obviously, to measure biological value, it's very hard to see the proportion of a protein, how much of it is incorporated into body proteins. Usually we use isotopes for this, so the protein will be labeled isotopically, and then we see in the body where radioactivity happens and how much radioactivity there is, and that tells you the biological value of a certain protein. Okay? How much protein should we eat? So the RDA for proteins is 0.8 grams per kilogram of body weight when you are maintaining your weight. So if you are working out, for example, it can go up to 2 grams per kilogram of protein. And so the recommended AMDR is 10 to 35% of total energy intake. Obviously, if you have a different condition, for example, a pregnant or lactating woman, they need higher amounts of protein. During growth, you need higher amounts of protein. Vegetarians need higher amounts of protein because their food is, or most of the time, lacks proper protein levels. We determine the amount of protein that we eat based on the nitrogen balance. So you need to have a nitrogen balance, nitrogen levels that are in balance. And so this is just basically the amount of nitrogen that you intake compared to the amount of energy, nitrogen that you excrete. Okay? If you're excreting more than taking, then that's a negative nitrogen balance, and if you're taking more than excreting, that's a positive nitrogen balance. When you are growing, you need a positive nitrogen balance because you need to take up more than you are excreting, because these proteins will basically be used as building blocks for the new cells you'll be making and whatnot. If you are in a starvation mode, or if you are not eating or whatnot, then your body will be mostly in a negative nitrogen. It has will have a negative nitrogen balance. Why? Because you're not consuming enough nitrogen and you are excreting a lot. Now, if you're not consuming a lot of nitrogen, how can you excrete more than you consume? Well, you're excreting the nitrogen that is broken down from your own proteins, because remember, the amino acid pool contains nitrogen or from the food and from breaking down your cells. And so if you have less intake and more excretion, that means it's coming from parts of your body, and so you're degrading your muscles. Okay, and this can lead obviously to very low energy, disease and whatnot. Adults need to maintain a healthy nitrogen balance, so that means you need to excrete as much as you consume. If you're not pregnant, obviously if you're pregnant, you need a positive nitrogen balance because you're not alone. There's another person that is basically requiring nitrogen as well. Okay. How many grams of proteins should a 65 kilo woman consume per day? Well, 65 times 0.8, I think it gives you 52. I think so, I forgot. 0.8 times 65, 52. Good. Why do athletes require more protein? Well, you probably think it's just to build muscle, but actually it's more than just muscle. So athletes do require more protein because they need to repair tissue that has been broken down while you're working out. That makes sense. You need more protein because it's used also as energy source. Remember gluconeogenesis and whatnot. You need also higher levels of hemoglobin, so you'll be making more hemoglobin to transport oxygen because athletes require higher levels of oxygen due to the physical activity they're doing. Gluconeogenesis, I just talked about it. Right, so I told you at the beginning of the lecture that too much protein can be harmful, right? Well, it can be harmful for many things. One of the reasons is heart disease. So if you're taking too much proteins, these proteins will be converted into fats, and that can lead to heart disease. That's the first thing, okay? So the extra proteins that you have in your body will be converted into lipids. Additionally, if your diet contains high levels of methionine and cysteine, then that can lead to bone loss. Okay, so this is a controversial information. There's research showing both sides of the argument, so there's still research happening on this. But methionine and cysteine contain sulfur. Sulfur can increase the acidity in your blood by producing sulfuric acid, and the body, the way it buffers this is by using calcium from your bone. Okay, so it will increase calcium excretion from the bone to kind of fix this issue. The main problem though is kidney disease. So if you go to your doctor, they will always give you tests, for example, for urea and whatnot, and this is just to see how good your kidney is at filtering your blood, specifically eliminating the nitrogen content from your blood, because you don't want to start accumulating ammonia and whatnot. And so this happens in a unit in your kidney called the nephron, which basically will take blood from the artery, filter it, and ship it back through a vein, the renal vein. Okay, so there's a renal artery that brings the blood, and then it gets filtered through the renal vein. Whatever urea, toxic compounds, and whatnot, they will leave as urine, right? And so when you take too much protein, you overload this process. Now, if you're healthy and you're young, for example, you're 30, 35, 40, this should be okay. But if you are a bit older, this can lead to a big problem by creating stones in your kidney, and those can lead to dialysis, right? So this is kind of dangerous, and you have to be careful with this. Usually this issue here happens with people that are working out. They take too much protein because they think that it's going to build muscles and whatnot, but actually it's very toxic for your kidney. Another issue with, one issue with drinking too many energy drinks have a lot of taurine in them. And because they have a lot of taurine in them, they have 2 grams, I think, of taurine. If you do it chronically, you can end up with this problem. And so the way you test this is by doing urine tests, and you look for proteins in urine. And if you find proteins in urine, that means this process is inefficient. Okay? Um, if you are a susceptible person, for example, people with diabetes, they also have a higher rate of kidney disease. Um, so nephrotic diabetes is a complication of diabetes. Let's just keep it at this. And so one way to help with the symptoms of nephrotic diabetes is to eat a lower protein diet to kind of not overload your kidneys, because your kidneys become less and less good at filtering stuff. Okay. Good protein sources, I mean, you know this. The higher you go here, the better, the worse they are. So nuts obviously are healthier than meats, whatnot. So this is just for you. There are two diseases that I want to talk about that you've probably seen on TV for countries that have famine. One of them is called marasmus. Marasmus is caused by an inadequate intake of nutrients, basically all the macronutrients and energy, which comes from those macronutrients. It usually happens right after childbirth. So when the child unfortunately does not have enough food because of being in a region where it's not very accessible, like here in Canada, for example, then what happens is that you don't get the building blocks for growth. That means you're not building a healthy heart. You have problems with brain development and learning. You have depressed metabolism. And it just makes sense, right? You don't have any building block to make whatever is necessary for growth. And so you end up having a phenotype like this. Now, I put this black thing here to hide the face of the kid because this is a picture I took from Google. If you take a look here, this is coming from the book, so they can deal with any problem that comes from this. But to respect this little child, just putting this here, okay? This, you're probably more aware of this phenotype than this one here. If you've looked at those, I mean, I used to watch them when I was younger, where they show kids that are not eating. So these kids get a belly like this, and this disease is called kwashiorkor, and it's basically loss of the phenotype is due to protein malnutrition, and so protein malnutrition, you know, meat is quite expensive compared to other things, and so obviously in countries where where some people lack access to meat, then they end up being in a state of malnutrition because their food groups are usually grains most of the time, not even vegetables. And so there is a weight loss that happens due to that. Because you don't have proteins, then you're going to start breaking down your own proteins to fix this issue, to increase the amino acid, the amino acids in the amino acid pool. So it's kind of a feedback loop, right? You don't have enough proteins, you break your own proteins to fill out the amino acid pool, which leads to making protein, but then you break down that protein, so you just keep losing muscle, okay? This leads to retarded growth and development. It can lead to edema. Edema is just a poor fluid balance, and so you'll see that they accumulate fluid in their in the interstitial space between cells, and this is basically full of fluid. The liver gets degraded, you lose appetite. Basically everything goes wrong, okay? Just simple like this. Question 5. Kwashiorkor, a form of protein malnutrition, is characterized by swelling throughout the body, particularly in the belly. Why does this occur? Well, it occurs because there are insufficient proteins in the bloodstream to draw fluid out of the tissue. And we'll see in a lecture about fluid balance the role of proteins into drawing fluids out of tissues. Okay, so proteins are important for drawing fluid out of the tissues. If you don't have enough proteins, well, you end up leaving the fluid in the tissues, and that leads to the edema that I just showed you. Okay. Another thing that can happen that is related to a disorder that can happen related to the proteins. We talked about phenylketonuria, right, which can cause brain damage. But there's another disease called sickle cell anemia, which is just a single amino acid change in the hemoglobin that leads to the hemoglobin to switch shape from this globular shape to a sickle shape, a shape like this. And this shape here travels less well in the blood vessels. Okay, because it travels less in the blood vessels, then you have a poor transport of oxygen, and so this can cause many problems, including vessel blockage, because these forms will kind of clump together, and it can lead to anemia as well. Okay, so phenyl, we talked about that. This is just another diagram to show you the normal, the one nucleotide change that leads to a change from a glutamine to a valine. And because you have this valine here, then that messes up the whole phenotype of the blood cells. And it does so by transforming the protein, the hemoglobin, from a globular shape to a kind of a long strand form. Okay. Another disease caused by protein disbalance, let's say, or mutation in proteins, is cystic fibrosis. Cystic fibrosis is a disease that is caused by a problem in your chloride ion channel. You have a mutation that makes it kind of in a closed form like this, and chloride cannot leave outside, cannot leave the cell. Cannot leave the cell. Because it cannot leave the cell, then the mucus that you have already outside the cell will be sticky. Chloride helps kind of turn the mucus into a less sticky form. So if you don't have the chloride ion going out, your mucus becomes more sticky. Now this is not dangerous in itself. What's dangerous is that you have mucus in your lungs and in your digestive system. And if those are sticky, then you'll have digestion problems and respiratory problems, because this will block the air from passing through. And if you are not, you know how important mucus is for digestion, right? We talked about this a lot. So if the mucus is sticky, that can cause a few problems down the road. Finally, if you are interested about, so this is just for you guys, I'm not expecting you to learn anything from this or to study it, but I've always been confused by the different types of vegetarians that exist. This is the list of all the types of diets that come from the vegetarian diet, what they can eat, and if there are comments about any problems that there are in these diets or any particularities of these diets. Okay, so you can just take a look at this. That's it, finally. Oh, it's so tiring to record 4 times in a row. You guys better be nice on on my evaluations. Anyways, so I'll see you on Tuesday.